In this section, you will see how pain is classified. Ari Kontos will be used as an example to describe how pain is detected or perceived in the body, and to show how pain signals are taken from around the body to the brain. Pain is normally classified into two main types: nociceptive, arising from being sensed by pain receptors called nociceptors, and neuropathic, neuro meaning nerve, and pathic meaning disease or condition. Nociceptive pain can be acute, such as touching a hot surface, or chronic, such as most back pain. In addition, it is possible to have a mixture of nociceptive and neuropathic pain. Sometimes the cause of the pain is not nociceptive or neuropathic, and such pain is termed idiopathic. Some types of chronic pain may be idiopathic. In this section, we will focus on nociceptive pain. Nociceptive pain arises from activation of pain neurons, that is, neurons that carry pain signals in response to a noxious stimulus that may be some sort of injury, disease, or inflammation. Nociceptive pain is caused by the activation of nociceptors. Nociceptors are receptors that respond to a number of stimuli. When they respond to chemicals, they are called chemoreceptors. Receptors that respond to temperature changes are called thermoreceptors, and receptors that respond to mechanical stimulation or movement are called mechanoreceptors. Individual nociceptors can sometimes respond to more than one type of stimuli. For example, there are nociceptors called transient receptor potential vanilloid type 1 or TRPV1 receptors. TRPV1 receptors exist all over the body and respond to heat, but also to the chemical capsaicin, which is the chemical in hot tasting foods like chili that causes a hot sensation when eaten. So eating foods containing chili causes a hot painful sensation because of the activation of TRPV1 receptors. Thus, TRPV1 receptors are both thermoreceptors because they respond to heat, and chemoreceptors because they respond to the chemical capsaicin. There is a whole family of TRP receptors, and the first of these nociceptors were only discovered in the late 1980s, and prior to this it was not known how nociceptive pain was detected. TRPV1 receptors are often found on neurons, and their activation then leads to activation of the neuron. The neuron sends electrical signals to the spinal cord, as we'll see in a moment. The effect of TRPV1 receptor activation is felt very quickly in order to alert a person to the source of the pain so that hopefully it can be alleviated. Chemoreceptors such as TRPV1 receptors can be activated by chemicals that come into contact with the skin, such as accidentally pouring acid onto the skin, or by chemicals produced in the body. When tissues become inflamed due to disease or injury, many different chemicals are released from the inflamed tissue. These chemicals activate various chemoreceptors in order to activate neurons that send painful messages to the brain. As mentioned earlier, there are nociceptors that are activated by movement called mechanoreceptors. There are a number of different types of structures that contain mechanoreceptors. Some structures detect light touch, whereas others are responsible for the detection of vibration or skin stretching. So they are nociceptors in the sense of detecting painful stimuli, but they can also sense non-painful sensations. So what about Ari's back pain? It may be the result of a strain in any of the interconnecting structures in the back, such as the tendons, muscles, and spinal discs. Straining of these structures can cause inflammation, which produces heat and chemicals that activate TRPV1 receptors or other similar receptor types. Ari's back pain may also be the result of mechanoreceptor stimulation. Often after an accident, there is structural damage that means that Mechanoreceptors are activated by simple movement of the body that would not normally cause pain. The precise way in which mechanoreceptors sense movement of tissue is yet to be completely identified. Mechanoreceptors may be similar in structure to TRPV1 receptors, and their activation may in fact involve the TRPV1 receptors as well. So when Ari feels pain in his back, it is nociceptors on neurons in the area that he feels the pain that are activated. These neurons are generally silent in that they fire much less frequently when they are not being stimulated. Once activated, these sensory neurons transmit electrical signals much more frequently from the site of the pain in the back to the spinal cord. Here, the sensory neurons release neurotransmitters that activate a secondary neuron that projects to the brain. This area in the spinal cord is an important target for drugs used to treat pain called analgesics, as we'll see in a later recording. These secondary neurons transmit pain information via electrical signals to various brain regions and eventually to a brain region called the somatosensory cortex. Let's now look at the somatosensory cortex in more detail. The somatosensory cortex is a strip of tissue across the top of the brain where pain, as well as touch and temperature, is actually felt. Each part of the somatosensory cortex represents a different part of the body. This is perhaps better represented by this model called the sensory homunculus. This shows what a person's body would look like if each part grew in proportion to the area of the somatosensory cortex concerned with its sensory perception. This highlights how some parts of the body are very sensitive, such as the hands and lips, and how other parts of the body are less sensitive, such as the arms and the legs. So to recap, you've seen how pain is classified, how pain is detected, how pain signals are taken from around the body to the brain, and how this pain is perceived in people experiencing pain like Ari.